When BACHBERRY was proposed and when it started, it was clear to us that it was a very ambitious process.
Basically, there's a large body of evidence behind bioactivities and functionality of the components that are in berries.
Plants are producing these compounds to protect themselves, or even in the case of berries, they are a very large class of polyphenols, anthocyanins. They provide the colors they have.
It's a group of compounds with quite potent antioxidant properties. So that's one of the major roles, but also they have antibacterial and anti-inflammatory properties.
You're looking at things like neuroprotection for things like Alzheimer's, Parkinson's, cardiovascular disease, things like cancer. Basically the three big diseases in the developed world at the moment.
So if we could have the entire population of the planet to eat enough quantity of berries, to have a good supply of these phenolic compounds, we would need an infinite planet to grow an infinite number of plants. So as that is not possible, it becomes essential that we can produce these very important compounds inside an industrial environment so that you can increase productivity without the need of so many resources.
So the way to do that is not to take them from the fruit, but figure out their biosynthetic pathway, lift the pathway in genes, and drop them into something that will only make that one compound. And this is what you would call an industrial biotech approach.
St. Petersburg in Russia. Right here in the city centre, scientists at the Vavilov Institute are safeguarding a very special treasure, the so-called Vavilov collection. The Vavilov collection is a collection of agricultural plants and crops, one of the largest worldwide, and maybe the most important of its kind. The collection is the outcome of a series of expeditions undertaken by Nikolai Vavilov, a famous botanist in the early 20th century. Vavilov traveled the world with a clear goal. As a Soviet botanist, his main focus was on crop diversity. He wanted to have as many varieties possible in order to develop new, improved crops and therefore help secure food supply for the Soviet Union. By collecting many varieties, Vavilov could trace back most of today's crops to the so-called centers of origin, places where the natural ancestors of our present agricultural plants can be found up until today. Being a scientist, not a politician, Vavilov preferred the ideas of Charles Darwin over socialist botanist theories, a preference that landed him in prison where he died in 1943. Nevertheless, his collection survived, was preserved during the Second World War, and is utilized to this day by botanists and seed developers around the globe. In order to keep the seeds germinable, some of them have to be frozen at very low temperatures, while others need to be replanted from time to time. Thanks to Russia's extensive landmass, Vavilov was able to establish subsidiaries of his institute covering the climate zones necessary to regrow his accessions. The Pavlovsk experimental station on the outskirts of St. Petersburg is one of the places where some of these accessions are grown.
The land that belongs to the experimental station is about 500 hectares. We have wild species, old cultivar that we conserve since 1926, and modern cultivars, and we have material, plant material from 17 different countries.
Taking part in the BACHBERRY project, the living plant collection of the Institute delivers berry samples to the European partners, therefore donating the basic material for further research. Today, Artem Sorokin is collecting samples from a berry called Lonicera.
We want to know what bioactive compounds we have and how we can use it. I believe that we will have very interesting results.
Filled into special bags and deep frozen, the selected berries will be ready for shipping to the project partners. Dundee in Scotland. Here at the James Hutton Institute, Derek Stewart is busy with a lot of berries he receives from partners around the world. Derek's institute has a central function within the BACHBERRY project.
We produce extracts, we analyse extracts, and we send out standardised extracts to the other partners to test. So we coordinate the fruit coming in. Now the fruit generally comes in as frozen material because it's safer, it doesn't decay. When it comes into us, we'll log it into databases, so we've got a good record, we can track everything. And then we go through a procedure of, we have a standardised extraction protocol that's been developed to capture as many different chemicals within the fruit as we can.
Today, Alexandra Foito is going to analyse the chemistry in one of the berry samples they received.
Well, we have a good idea what it might be there in terms of classes of compounds, but exactly what it is, their decoration, we don't know for sure, especially because a lot of these samples have never been analyzed before, so we're doing something completely new over here. I've received some Lonicera samples from Russia, and at the moment I'm going to extract these berries.
In order to execute the analysis, the berries have to be processed into a liquid state. This so-called extraction procedure is carried out in a standardized way for each berry. This makes it possible to compare the chemical compositions at a later stage.
Berries have a lot of sugars, organic acids, but more important, they have these compounds called polyphenols, which are our target compounds for this project, and this is what we're trying to study.
The different molecules of the berry are detected by the use of heavy machinery.
This is one of our mass spectrometers. So what we're doing over here, we put our samples in here. This is the auto sampler, and the machine just injects a little bit of our sample that goes into a column over here that we use to separate compounds. So anything that's present in our berries, we try to get it separated. And then after that, we'll just collect some spectrometer data, and then it goes over here into the mass spectrometer, where we look for the masses of our compounds. So this is the kind of information that we get from our mass spectrometer. Sometimes for compounds are very abundant and very well described, it's easy to identify them, but sometimes we need to look further and see how the compounds break when we try to fragment them. And that gives you information regarding the structure, and that helps us identifying some of these unknowns.
Each peak represents the mass of a molecule that was detected. To find out which molecule it really is can be a puzzling job.
It's just piecing all the pieces of information together, and eventually, in the end, we come up with a true identification of what's really in the sample.
I think before the project was actually constructed, there was a lot of discussion as to the target fruits, and it was clear that the ones we have chosen show, according to literature, a wide range of diversity with regard to their chemistries. And if their chemistries are diverse, potentially their usefulness of these chemistries will be diverse as well.
Claudia Nunos works at EBET, a research facility near Lisbon, Portugal. Her research is dedicated to mental health. Therefore, berries could play an important role, as Claudia receives promising extracts from Derek's lab in Dundee.
We get a total extract of polyphenols from a berry.
It is assumed that the polyphenols from berries could positively influence Parkinson's disease.
Now we want to understand which is the compound that is more promising for that specific disease.
Don't be afraid.
The phenolic compounds are tested on yeast, but not an ordinary one. It's a genetically engineered yeast that suffers from Parkinson's disease just like a human brain would. Therefore, it is called humanized yeast.
What we have is yeast that is expressing human proteins. It's not a protein that is expressed naturally in the yeast. We transform the yeast to express this specific protein that is a hallmark of the disease that we want to study.
By producing the proteins that trigger the disease, the yeast itself is of a self-destructive nature.
The fact that the yeast is expressing this protein, it becomes toxic to the yeast, so they are not growing so well.
By this, the growth of the yeast becomes an indicator. If the berry extract can prevent the protein from killing the yeast, it may also save our brain cells from the same protein. Different growth curves, therefore, prove the efficacy of a substance.
So, for instance, we have a growth curve, and we see the difference of the growing due to the fact of putting the polyphenol in yeast. And the ones that were improving the growing of the yeast, we decided to continue study which type of mechanism is behind this protective effect that we see in the growing.
Understanding the way the compounds within the berry extract prevent the Parkinson's disease proteins from destroying cells is the precondition for developing a drug. In any case, yeast would be just the first step on a long journey.
We go to the human cells and confirm, and of course we are thinking about a pharmaceutical. We need to go for clinical trials, and even before the clinical trials we need to do tests in animal trials and other confirming everything, and will take longer to reach to the consumer, of course.
For a long time, berries have been known for their viability as a food colorant. This lab in Montpellier, France, is specialized in the development of natural food colorants, a highly demanded product in today's food and beverage industry.
Consumers are more and more aware of what they are consuming, and they want more natural food and also more natural beverages, also concerning the ingredients and the colors.
Finding a color pigment within a plant does not automatically mean it can be used. There are many requirements a pigment has to fulfill in order to be used in industry. An essential property is the availability of the pigment in large amounts and a constant, stable supply. This is where the concept of BACHBERRY could make a difference. Producing natural colorants in a bacteria can guarantee a constant, stable harvest, and therefore unlimited scalability and supply.
The University of Groningen supplied a different type of bacterial extracts. They managed to introduce genetical material from berries inside the bacterial cells. I evaluated the color content, the shade, and then I gave the samples to one of my colleagues in the beverage team, and they evaluated the behavior of the bacterial extract in soft drinks applications.
Testing the pigment as colorant for beverages starts with a standardized beverage.
We use a standard recipe we call internally standard soft drinks. So we have always the same chemical and physical parameters, like the Brix and the pH of the drink.
The BACHBERRY pigment is mixed with the liquid, making it comparable to other already existing colorants.
we compare with standard colors we have or other colors we have in our range, and we will also measure the color to identify what are the differences.
For the industry, it is important that a pigment is able to withstand the harsh conditions of transport and shelf life. A professional pigment has to be tough.
So we put the bottles in the spot chamber. They will stay here 4 weeks long, and we will check the bottles every week. We will make pictures. We will control if they are fading or if the color is shifting over time. This is a kind of accelerated test. The bottles are exposed to light with these two spots, giving the same spectrum as the sun. And they are exposed during the night because it's very, very strong, 12 hours a day. Here I will put the bottles in the heat cabinet, and we will test their robustness towards heat. We will leave them 4 weeks long, and we will check them every week as well.
After testing, the colorants are compared. That comparison needs something more precise than a human eye, the color meter. It is able to detect even the smallest differences and give them numeric values.
The lightness, the chroma, so this is the intensity of the color, and the hue of the color. And by comparing with the second sample, the software will be able to calculate the difference between the two samples. We have actually the first color compared to the second one, so we can see that it's really, really close. And here we have the figures of the lightness, the chroma, and the hue. So we can say that the color of the second sample is slightly more bluish. The intensity is slightly higher for the second sample, and the lightness is lower.
If a pigment is detected that meets the needs of food production, it is still a long way to bring it to the market, especially when berry extracts are no longer produced by berries.
We have some compounds, for example, the beta-carotene compounds. They are produced with microorganisms, so this is something possible. But this naturally, these bacteria, they are producing these pigments. In the framework of BACHBERRY, it's different because we are changing the genetical material of the microorganism. So in terms of legislation, it's more complicated.
Nuno Faria from Biotempo in Braga, Portugal, is engaged in industrial fermentation.
So a fermentation process is a process where you have a vessel, you put a liquid inside with some nutrients and other chemicals, then you add some microorganisms, and the microorganisms convert some of the chemicals in the liquid to interesting chemicals for several purposes.
Nuno takes part in the BACHBERRY project for a good reason.
When you think planet-wise, so if you think that the entire population on the planet needs more phenolic compounds, you would need a lot of landfills to grow all these berries, and you would deviate arable land from the production of other food that is also necessary to feed the world population.
When it comes to the production of chemical compounds, bacteria in a fermenter have big advantages over plants.
A fermenter that occupies a space of 2 by 2 meters would have about 10,000 liter capacity. So you could have, perhaps you could have in 1 liter of fermentation the capacity to produce the same amount as one berry tree. So in a 10,000 liter fermentation, that would be 10,000 berry trees, and this would occupy a space of 2 by 2 of land that does not need to be arable.
Moreover, fermentation can happen anywhere.
So you can do it in stages, for instance in a building in several stages. You don't need the sunshine. It can be done in the desert, in the North Pole, wherever you can do a building. And so that is the difference.
Once the genetic information is transferred from the berries into producer bacteria, the BACHBERRY compounds can be produced independently, saving arable land for food production. As other project partners were able to engineer the polyphenol-producing bacteria, it's up to Nuno to raise them well.
They have done experiments in flasks, in 100mm flasks. Now, when we moved to a fermenter, we changed to a vessel that already has a mechanical agitation, that has an air inflow, that has controlled pH, controlled oxygen, controlled temperature, and also where you can change the operation strategies. And all this could have an impact on the fermentation. It could be positive, it could be negative. So doing this upscale is not straightforward. There are a certain number of variables that have been controlled and that have to be assessed, but that's our work, that's what we do.
As testing compounds from berries, engineering bacteria and experiments on fermentation are continuing, BACHBERRY already achieved an important goal.
The most valuable thing that we have now, and we will certainly have at the end of the project, is the tools to develop these technologies. If the project would continue, or if a new project would come up in the sequence, I think now we would be a lot more efficient in identifying a lot more of these target compounds and developing the industrial processes to produce them. In the case of the BACHBERRY, if we identify one target compound and we are able to do it at the fermenter scale, then it will be extremely successful.